We will begin with the second part of the day. First, I want to introduce, we have the full lineup of speakers: Professor Dengel, Professor Dumit, Professor Loncharet. Welcome to Argentina. And we are so happy to have you here. And we can't wait to hear your talks in the following days. Next, I have the pleasure to introduce Dr. Eduardo Ceccarelli. I know he's my boss, but I really pleasure to introduce him.
Should be for you a pleasure to introduce me.
Eduardo is a biochemist. He's a researcher at CONICET, and right now he's also the head of the mass spectrometry unit at IBR, as I told you in the morning, the newly funded unit that we are very proud of. And continuing with the first day of basic concepts in mass spectrometry and in proteomics in particular, Eduardo will talk about sample preparation. That is a very, very important step in the workflow of proteomics. Thank you, Eduardo, for being here.
Thank you. I'd like to thank to Nico and Germán for the huge amount of work they have done to organize this course, and everyone here, because the course is wonderful if you have students and people that are interested in the course. As a matter of fact, I will take only 5 minutes for a personal reference. I did my first proteomic experiment in 1983. So even the word proteomics didn't exist in those times. We constructed our own two-dimensional equipment for electrophoresis, and we pick up a protein that bound to another protein, and a factor that was in the membrane and was holding the reductase, that was the protein that we were studying in those times. We didn't know that we were doing proteomics, you know. We were looking for interactors. So, and the things have changed a lot. So I will try to talk today about protein preparation, but the very basic things of protein preparation. So don't feel offended if something that I presented is very well known for you. And I will try to talk about some of the precautions that you have to maintain in your lab for. This is only a reference. If you look for proteomics, proteomics is a growing up field, a huge field. In the references, you know, only in this year you have like 15,000 references, and 300 of those refer to sample preparation. Of course, I did a keyword search, so probably are even more. So that means that protein preparation is an important aspect of proteomics. If you're thinking to get good results, you need to start with a good sample. The proteomics stand in 3 important aspects of the technology. Your sample, the quality of your sample, your instrumentation, you know, you need to have a good equipment, and then you need to have software and people that understand the software to extract from the data that you get the important information. So when we talk about what is a good sample, what kind of things we're thinking about? Well, the first things that not always is true is that the sample represent the protein composition of the tissue we are studying. This is not an obvious aspect. Sometimes we want to get all the proteins out of the tissue or the cells, but we only have a fraction, and we don't analyze exactly what we want to see or to investigate in our sample. The other important thing in sample preparation is the sample processing in a way that you have a reproducible results, so that you can give replicas, and this replication is exactly the same, and that you don't have losses or enrichment of protein that should not control. That means, of course, you will see along the course that you probably in some situation you need to increase or deplete the sample of some protein, but the idea is this is a controlled process, not something that you that happened by chance. And you need to know that your sample does not have impurities. That means that you don't have things that shouldn't be there. And the other important things for people that run a facility and for the owner of any equipment is that the sample does not damage the equipment. That is something that can easily occur. Well, we need to know what do we want the sample for, what kind of studies we are going to do. I will mainly concentrate in the very basic sample preparation of bottom-up proteomics, and we are not going to talk today about top-down proteomics. So the first important aspect that we have to check, to think, and to planify is to know what are our source of protein for analysis. And each sample has its own particularities. Each sample has, each tissue or cell has a problematic aspect that should be keep in mind. For example, if you are working with cells, you have to know that contamination is a frequent problem, and with bacteria and yeast, with virus, with other cell types, you have to check that you have a genomic and phenotypic instability, and that the cell culture composition alter your quality and the protein content of your cells. This is the same for tissue culture. And organs, especially when we are talking with human organs, up to now, there are, let's say, less penetration in analysis. Is more difficult to work with, and but you will find approaches to deal with this kind of tissue. In the case of eukaryotic secretomes, you have to keep in mind that you have the protein and membranes, that you have different vesicles and particles and different type of bodies that appear in different stage of the culture of the cell. So it's important to control perfectly all these aspects. And if you're working with urine, you have a population of proteins in the sediment and soluble and urinary exosomes that can be fractionated previously to the processing of the sample. And that is the same for cerebrospinal fluid, but here you have the problem that normally you get blood contamination during extraction, practically in 80% of the cases, and that you have a high dynamic range of protein concentration that is the same that happened with the blood itself. That I don't find the blood here. For some reason is missed, but you have samples in which you have a high dynamic range of protein concentrations. So some of a protein, as Pia mentioned, can cover the possibility to analyze and to investigate other proteins. And in the case of microbial cells, we already have multiple protocols, and probably is one of the fields that is had been increasing quality for protein preparation. In the case of culture media, you have to take it into account that you have a lot of secreted protein, but you have also proteins that are coming from the lysis of the cell, and also the proteins of the media where you grow your cells. And saliva is 30% of blood proteins, and you have an inherent instability by proteolysis of a sample. And in the case of tumor cells, circulating tumor cells, it's an interesting field that is growing in the last few years because you can perform analysis without without the need to take tissues of tumors. And there is a protocol for purification of the circulating tumor cells and increase the quality of the sample. In the case of plant tissue, of course, you had to take into account that your growing environment, your recollection, your tissue classification is really important for the quality of the sample that you have. As you know, the sample preparation, as you already talked, Pia, can be subjected to different steps like metabolic labeling. These samples coming from cell or tissue could be lysed and the protein extracted, and you should quantify in some way the protein, and you can even deplete it from samples with a high quantity of few proteins, the abundant protein, and deplete it, the abundant protein like albumin or immunoglobulins in the serum. And then you can even immunoprecipitate or activate or make active site labeling for enrichment of the sample. Of course, the enrichment can be done previous. I'm not talking deeply on that because there will be talks covering this subject, but you can make an enrichment of a sample previous the digestion. You can make a peptide enrichment, or you can even precipitate the sample, the sample and only allow the analysis of intact proteins. Well, basically the protocol that you will employ for any protein preparation is lysis, fractionation, depletion if it's necessary, and protein determination of the quantity. Then a purification that can be done by gels in solution or in any filter-assisted technique, and then fractionation and cleanup and the mass spec of the sample. Basically, the SDS-PAGE sample preparation and proteolysis is depicted here in this scheme. You have the protein extracted. You can denature, redact it, and make the alkylation of a protein. Then you can run a SDS-PAGE, excise the band, which is detected by staining, and then the band is subjected to in-gel digestion and peptide extraction, and then you can add a purification step to get rid of contaminations in this process, or you can make the SDS gel electrophoresis, then to cut the band of the gel, and then run the denaturation, reduction, and alkylation in gel that will block the formation of disulfide bonds and will add 57 Daltons to each cysteine that is modified. And then they proceed to the in-gel digestion and peptide extraction. The idea to run a gel for purification is, well, we normally run in the lab full separated gels where we have the band resolved by the gel, but the idea of using a gel for protein purification is to run a short separated gel where all the proteins only run 5 to 10 millimeters on the gel. Normally, this is a gel, SDS gel of 10 or 12% polyacrylamide concentration, and this is a step that most of the polymers that are not that impurify the sample are purified. Among them, in polyethylene glycol, that is not that polyethylene glycol does not run with the protein sample, and this piece of gel is structured. Here you can see the different pictures of gels. This is the original method, and this is a gel running in our lab for protein purification. This is a guy that shows how all his samples are identical in gels for purification, and this is the way that you should cut the gel. In this way, you are recovering all the proteins that you have in the sample, and all the proteins have been purified, and you know that there is some unwanted material that will remain in the loading slot. And it's quite important in this process not to contaminate the gel, so you should make the cut of the gel over a glass that is perfectly clean. It's not advisable to use plastics or plastic sheets or any other support. You have to use a spatula and scalpel that will help you to cut the pieces of the gel, and you have to leave the pieces in a way that the amount of buffer needed for cleaning and proteolysis is reduced to a minimum. So the gel slice in the epidermal tube normally is destained with the destaining solution, and the idea is that to remove all the dye from the protein band, so it's advisable not to stain a lot the gel in a way that you can remove the staining material, the dye that is bound to the protein. Because this dye is toxic for your columns in the mass spec equipment, then you will reduce the protein with any agent for reduction, and this protein can or not can be washed. And sometimes you can directly use an excess of iodoacetamide, and this will block the sulfhydryl groups, and then the gel pieces are dehydrated, and to clean the pieces, and then you will swell the gel pieces adding the trypsin solution. The gel fragments, of course, should be immersed with the solution of trypsin. After incubation, depending on the quality of your trypsin and the way that you are doing the digestion, this digestion can be checked for quality, but normally it's an overnight process unless you can use another helping protease, LysC. I will talk about LysC in my talk of lip mass spectrometry. And then you directly remove the solution. You have here practically all your sample is in this solution, and then you can extract twice if you want with the acetonitrile and with formic acid, trying to recover in the charge and the hydrophobic peptides that can still remain in the pieces of gel. This is the sample that should be dry and injected to the orbit trap. There are different systems to prepare samples. In an article that was compared the quality of the three different systems in gel, S-trap and SP3 magnetic bed purification. And basically, the system is you get the sample, you get the extract of your protein as whatever the way did you use, maybe using a beating machine or any extract that you know how to prepare. The proteins then are processed with different system. Let's see how the other system work. The system, the S-trap, have a membrane of derivatized silica. The lysis and solubilization of protein is made in SDS because we can clean perfectly the sample in these columns. Proteins denature by acidification and high concentration of methanol are solubilized, reduced, and alkylated, and then this sample is put in the S-trap column. The solution that is used to keep the protein the solution that avoid the protein to go through the membrane, you can say there is a solution that make a kind of colloidal particle of the protein. Not only the protein is doesn't go through the membrane because the shape and the quality of the membrane and the pore of the membrane, but also because the proteins are in some way precipitated in solution. So that's allowed to clean the protein, and then in this condition is incubated and digested. In some cases, it's a good idea to add the surfactant to facilitate the digestion of the protein, and then the peptide digested are changing the buffer, and this allows the peptides to go through the membrane and to be recovered in an Eppendorf tube, and this is the tube that is dried and injected in the equipment. So the other methodology is the magnetic particles. What is good to use is a mix of more hydrophilic and more hydrophobic surface particles, but all of them with the carboxylate. These particles hold proteins when you get a high concentration of organic solvents. So you can bind the protein to the particles. You can wash detergent, chaotropos, salt, and other chemicals if necessary. And then you can do two things, or you can directly take the protein of the peptides out, or to make all the reaction in the same tube. So you get the protein, you induce binding, you clean the contaminants, and then you can do two different steps. Or you take out the protein for any other step of like gel separation, affinity enrichment, or chemical labeling, or enzyme digestion, or you make the enzyme digestion in solid phase, and you elute the peptides for analysis. These guys have compared a mixed sample of E. coli, yeast, and Bacillus subtilis that are mixed all together, and they analyzed the time of each optimized method for them, and then the number of peptides and proteins that they identify. Well, a couple of conclusions are interesting in this work is that the SP3 system is the one that allowed the higher number of peptides identified and the higher number of proteins identified. But something that it called me attention is the fact that depending of the method you use, you have a population of peptides that are identified no matter which method you use for purifying the protein. But there are peptides that are only identified using one or other purification system. That means that in some cases there are peptides that for physicochemical characteristics are purified better in one system than another, but it's not that I'm trying to sell the SP3 particles, but it's the system that for them is the quicker and the more effective system for purification of proteins and peptides. Well, this is the same of other just recently appeared paper checking an identical protocol of extraction, a protein extraction of a plant sample, and then the downstream purification of protein and peptide identification for the different systems and with different times of protein trypsin digestion, and seems to be that the sp3 carboxy is the more effective system for peptide purification. You can have problems where your proteomic sample preparation. You can have a low quantity after your step of purification. That is a problem because normally you will identify the more abundant peptides and proteins, and you probably will lose important information. Another thing that can occur is the sample modification, and another problem is the contamination of the protein sample. Checking a database of something like 10 million proteins, this is not so new as 2017, but you have that carbamylation is the more important problem in unwanted sample modification, and deaminated peptides is also another problem. The inconvenient is that if you check modified peptides can be responsible for 20 to 50% of the false positive identification in deep proteomic data set. That means you get a peptide modified and you confuse the identification, or you lose the capability to identify this peptide. So from characteristic of conflicting matches from accumulated proteomic data from HAK 293 cells, they found that 22% are carbamylated peptides, and methylated, deaminated, and other problems are coming from peptide modifications. The thing is that the way that we treat our sample can favor or not some of these problems. All samples, bad treated samples that are not properly manipulated, can give these kind of problems, and this may of course, a carbamylated peptide won't run in the same position that the unmodified peptide, and this peptide won't be identified in our run. So carbamylation comes mainly from urea treatment. The urea forms isocyanic acid and will react with primary amines of proteins, but also, in lesser extent, with epsilon amino groups of lysine. It adds a mass of 43 Daltons to the peptide, each reaction, but sometimes you can have more than one reaction per peptide, and fractions of reactions, so that these peptides are lost. And excuse me, one of the problems is the formation of isocyanic acid is spontaneous in urea, and after you prepare your solution of urea, the isocyanic acid start to form and is accumulated. So all solution of urea always has isocyanic acid, and if you treat your sample at temperatures higher than 37 degrees, you will have modification of your sample. And the modification of the sample can be previously to the steps of protein purification, or during the step of protein purification if you use urea in some of the step of the purification. Another problem of sample contamination is the common laboratory reagents and handling samples and the material equipment used. The most relevant problems of contamination is keratin contamination, other proteins used in the lab. Sometimes a proteomic lab coexists with the lab that is producing a huge amount of recombinant proteins. So you have a yeast extract, or you have a peptone in your lab, or you even have a huge amount of the express protein that is in the sink, and the material, glass material, is used in this way. And polyethylene glycol and Polysiloxanes. Keratins come from the skin and the air and you know, and the hair, and it is present in quantity in the laboratory dust. And normally you get always keratin. The people that run the proteomic analysis can try to decrease the keratin, but depending on the amount you have is how will affect your studies. Well, that's something that I have told. How to deal with keratin contamination? Any laboratory surface exposed to atmosphere for a few minutes will be contaminated. So you don't have to leave things around the table. So you have to keep all the places clean, cover, and seal. So you don't have to leave open gels waiting to be cut for hours over the bench. This means you have to work in a clean area. You have to use always protective items such gloves, masks, glasses. The idea is even use caps and overalls. And anything that is left to dry next to the laboratory sink basin where people sanitize their hands should be considered contaminated with keratin. So you don't have to run your hell and leave the glasses or your electrophoresis equipment near the place where the people clean their hands. And polyethylene glycol is highly distributed. It's normally strongly ionized and can generate a lot of signals. The problem is I have a repeat of 44 Daltons, so these kind of things you can see with high contaminated polyethylene glycol samples. It's hugely used in cosmetics, in detergents, in all these washing soaps that are for cosmetics. So it's better not to have these things in the lab. So not to use dishwasher detergents. It's normally used at high concentration. And the other problem is the siliconized surface or plastic polysiloxanes that also generate polymers of 76 repeats that are also detected in the equipment. Most of the system that I have already presented for purification samples will clean most of these polymers. So this is something that sounds quite obvious, but you know, you don't have to use wet wipes to clean your hands or the bench tops in your lab. The wet wipes that are selling commercially have a lot of compounds that are contamination for for the mass spec people. Do not leave material in the sink. You don't have to use glassware that was already used to prepare any solution with polyethylene glycol. And you have to use only solution in glassware. Not is a it's not a good idea if you have a flask that was used to prepare a solution that you will use to send all the way to the cleaning room and get back the glass, because this glass is clean. So you've finished with the solution. It's better to clean the bottle with some high quality water and use again the bottle, unless you have some contamination in the bottle. My advice is not to have dishwashing detergent inside the lab. And when you use disposable pipettes and other pipettes, you should try to use only for the mass spec project, and not to use or to share this kind of equipment with other projects that are high contaminating projects. In the case of polysiloxanes, you should always use HPLC mass spec grade solvents. You don't have to store organic solvents in plastic bottle, even if there are mixes with water or with other buffers, because you get the organic solvent dissolving polysiloxanes. The quality of the tubes that you use is important. Not all the tubes have the same amount of contaminated material, and avoid any kind of siliconized surface. This treatment generate polysiloxanes. For example, some people siliconize chromatographic columns for purified proteins, or you siliconize for running some kind of electrophoresis. This is not good to have it in the lab. That's all to have an idea of protein preparation. As Germán told me that you will have a place where you will get documents and reprints. So I will upload a series of methodologies and papers of methodologies for sample preparation. And you can also go to our page in the Institute, where we have posted some of the things that I mentioned here, and a detailed protocol for gel electrophoresis protein purification. So feel free to contact us if you have any question about these issues. Thanks.
Thank you, Eduardo. Great talk. Full of details. Questions for Eduardo?
Hi, Eduardo.
Hi.
Sí, ¿te escucha? Parece que no te escucha. Hi, Eduardo. I wanted to know how often this contamination occurs.
Well, let's say when the people is not aware of the way that you have to work, normally samples have some kind of contamination. Keratin is so common from people that are adapted to work in the biochemical lab, but it's not doing proteomics. So the person that work in in mass spec, it's like in other times the people that working with RNA, that you start to know that if you are snoopy with your sample, then you don't get a good result. So, but keratins, always you will have some kind of keratin in the sample. So we live with the keratin in the environment, and you will see always keratin. The problem is when you get such amount of keratin that your studies keratin is not any other thing. So it's not let's say if you are cautious when you work, you shouldn't have problems with the contamination.
Thank you. Hi. So you introduced the protein sequence of keratin, albumin, and all the contaminations on the database to analyze the proteins, and you take it into account, or how you do that?
Well, in some cases you can't reduce the amount of protein that go into the equipment, but you can't not to analyze it, because when you are working with a serum, you should do it. You can avoid the signal, or you can avoid interpretation with your software. But in the case that you have a high signal, the signal will be on the equipment. You can manipulate your signals up to some range when you when the contamination is you can say, well, I will omit keratins of my analysis because it's not only keratin.
Yeah, if I can add one comment, yes, in the databases we can include the database of your organism, and there is another database that is very common in the facility of proteomics called the CRAP. Trash that is available online, and there it has a list of keratins, casein, GFP, a lot of common contaminants, and we also include that in the analysis. And as Eduardo said, we always find keratins in every sample. But yes, the answer is we also add this database to your database from your model organism. Okay. And we always find keratins.
I work on the security cabin of level 2. It's okay to prepare the samples under this cabin, or?
I don't think that it's necessary to have some kind of special facility.
Okay.
It's not the way that you contaminate, like something that is coming out of your sample. Well, depending on what kind of organism you are working with. But in that case, you have to take the caution for the organism.
Okay.
But in sometimes in some samples coming from human, the keratin may be an important protein to follow. Maybe the changes in the level of keratin may have a meaning for your studies. It's not always in that way, but it could be. So in that case, you have to be extremely clean to run this kind of samples.
Thank you.
When you work with plants or E. coli, you know that you don't have to see keratins there, but you are running a tissue. You are running a tumor of hepatic tumor. You don't know if the keratin may be important for the. In that case, it's even more problematic, the contamination.
Thank you, Eduardo. Beautiful presentation. I have a question. You show us 3 methods. One was SP3,
Sí.
S-trap, and the other one was in gel.
Sí.
And with SP3, you have the large amount of proteins, and also was the fastest?
The large amount of identify proteins
Identify
and
proteins.
the large amount of peptides, identified peptides.
Yes.
In this paper, they assume 2 peptides for protein identification. That is something that you can. This is a parameter that you choose to have. They say, well, I identify a protein. For me, a protein that is that was identified is a protein that I identified 2 peptides. That's what these guys have done.
Okay, and you think that is true for every kind of sample?
Well,
or
they I don't know. There are a number of concurrent peptides, the papers that the amount of peptide with sp3 are higher, not only for plant tissue, also for other tissues. And these guys have a mix different cultures of bacteria and yeast, and they identify proteins coming from bacteria and from yeast, and they found that there is the system seems to be better. One of the problems, of course, one of the problems is the cost of the column. An S-trap column is like $9 in United States or in Europe, so you have to add the cost of having the column here. But it's something that is important to think about the proteomic analysis. It's something that is it's fast to put the sample in the equipment and to run and to have data. But you can have rubbish of this data if you don't think of what to do in the experiment. So sometimes the cost of the experiments is not this step of protein preparation, is what do you want to have, what kind of data you want to get, and how long take to you to get the sample for that proteomic experiment. So I think to plan perfectly, as Pia told you, to perfectly plan and to study your tissue, your cell, your system, what you want to see, what is your hypothesis, working hypothesis, is really important. Another way, you don't get good results in proteomic experiment. You have to know perfectly what kind of question I are trying to answer and how to extract the data to answer these questions. So this is a work that should be done previously, and you have to analyze what is the tissue, what you want to do, and how to do it before you put the sample in the equipment. So I think this is really an important point and when you reach to the sample preparation, you should have the best possible sample to put in the equipment.
Thank you.
Thank you.